Hello everyone and welcome to this first bio webinar of the new season of events organized by the Finnish Society for Bioinformatics. Bio webinars are bi-monthly activities where we invite both junior and senior researchers to present their recent scientific works. My name is Nushi, and together with Xin Lai and Julia Mathlin, we take care of these events on behalf of the Finnish Society for Bioinformatics. The bio webinars are recorded and you can access them on the webpage of the Finnish Society for Bioinformatics. Our speaker for today is Arber Qoku. He is a doctoral candidate in computer science affiliated with the Goethe University, Frankfurt, and the German Cancer Research Center. His research focus includes developing statistically sound and computationally efficient latent variable models for comprehensible and scalable integration of multi-omics data. Arber will present us today the software package MOFA-FLEX that is a factor model language for integrating omics data with prior knowledge, which he developed with his team. Thank you for accepting the invitation to have the presentation, Arber, and you can proceed with your talk.
Thank you, Elio, for the introduction, and thanks everyone for participating and listening to my talk. So, as Elio said, I'm a PhD student at the German Cancer Research Center, based in Frankfurt, and I'm part of a bioinformatics research lab called the MLO lab, which stands for Machine Learning in Oncology. And the focus of my research and the main topic of this presentation is the development and the study of multi-omics data. So essentially what we try to do is find an interface between having multi-omics data, developing the right methods that are efficient, computationally feasible, and statistically sound, and at the same time integrate some existing prior knowledge from, let's say, previous studies. And what we'd like to do is combine all these three main things in order to interpret the multi-omics data that we have, try to explain different sources of variation, hopefully by integrating the prior knowledge, as well as then move one step further and make sort of predictions like disease outcomes or survival analysis. So we can start our discussion with a very general multi-omics problem. So let's say we are given a set of matrices that represent expressions from different omic layers. So the rows represent samples, which can be patients or cells, and the columns represent the omic-specific features, which can be genes, proteins, mutations, and so on. When we perform a matrix factorization, similar to a PCA, where we infer two main components. First, we learn a common latent space that describes the data in a much lower dimensional space. And second, we infer a set of linear mappings from this joint latent space to the observed data. And these are called the factor loadings. And they describe each latent factor in terms of the, let's say, the weighted features of this corresponding vector. And you can think of it, as I said, as a sort of decomposition in the sense of a PCA or a factor analysis and so on. This is closely resembling a group factor analysis. Typically, the factor loadings that are represented here, they are passed to a sort of a gene set enrichment analysis tool to kind of understand the biological theme behind each factor after training. What we propose is taking this vast public domain knowledge, such as the gene set collections, and provide it to the model as a training context. And this context then should guide the model to infer latent factors that align with our understanding of the data. So for instance, if we're, let's say, interested in quantifying whether a subgroup of cancer cells exhibits high levels of hypoxia, let's say we have a bunch of patients with lung cancer, we can provide a set of genes that are triggered or activated in a very hypoxic environment, and then let the model quantify the levels of hypoxia for each sample in this case for each patient. And then what we can do is then we can stratify patients that exhibit low hypoxia, medium hypoxia, and high hypoxia. And eventually, this might lead to particular patients having a lower or a higher survival rate based on this particular biological pathway. And we would like to do this in parallel for hundreds of such different biological pathways. There are two aspects that I'd like to point out about our method. One, we accommodate noisy gene sets. That is, existing genes in this particular gene set collections can be turned off, or new genes from outside of this gene set can be added or introduced to the gene sets if justified by the data. And then second, the model works quite well when even informing a single modality. And in this case, here in this example, we inform the RNA only because it consists mostly of genes compared to a proteome or a mutation modality. Then the shared factors allow the prior information to flow to the uninformed views. Then after training, we may then proceed with a downstream analysis, such as imputing any missingness in the data. We can investigate the different factor composition. We can visualize our samples in terms of now pathway scores instead of gene scores. We can remove factors that might correlate strongly with unwanted sources of variation, like age, sex, or different technical artifacts like batch effect, and try to kind of focus mostly on the let's say the biological insights from this data set. I have here an example on the well-known Cancer Genome Atlas data. So to showcase our approach, we applied our method on the TCGA dataset of 11,000 samples spanning across 33 different cancer types across 4 different omic layers. So we have DNA methylation, RNA, microRNA, and RPPA, which is a proteome. And the model was informed from around 360 gene set annotations. They all come from online databases like the Hallmark, Reactome, and the Keck database, which is readily available online. Since our method performs a dimensionality reduction, as I said, the joint latent space, we can apply out-of-the-box visualization tools to kind of gauge whether the model can really separate disease subtypes meaningfully, which can be seen here by the t-SNE subplot. Then we can, so this can be seen as the kind of a global overview. Then we can move one layer deeper and perform statistical tests to pinpoint, let's say, the most relevant pathway or latent factor informed by the pathway for each cancer subtype. And among others, we see melanogenesis, which is quite relevant for skin cancer patients, and androgen response as highly relevant for patients with prostate cancer. Finally, we can then investigate the composition of the factors informed by, of the particular factor informed by the androgen response. The black dots represent genes that were present in the gene set before training, and the red dots represent genes that have been inferred or added into the gene set during training because it was, let's say, justified by the data. And in the x-axis, we have the corresponding value for the factor loading, where higher means more important. There are 2 things that we notice about this particular factor. One is that the informed factor, or the informed modality, is refined in such a way that it aligns with existing literature. So there's ACPP, STEAP1, SLC, and these are all well-known biomarkers in prostate cancer patients. And the model was not informed by these particular markers before, but based on the correlation structure, they have been added, let's say, to the gene set a posteriori after training. Second, because this factor is shared, we observed that in the uninformed view, which is the proteome, it ranks CHEK1 and BCL2 quite highly. both being from the literature oncoprotein targets of having clinical relevancy. And this implicit information flow across shared vectors works quite well because of the correlation structure. And in most cases, it can hint towards, let's say, novel feature sets in other omic layers where, let's say, this prior knowledge is less present and less common, like protein sets. So that was the kind of the main idea of how to integrate kind of prior knowledge into this multi-omic integration methods. What we then try to do is take different approaches in the community that perform multi-omic data integration and try to bring everything together in one unified framework, which we call MOFA-FLEX. And similarly to the previous method, which was focused mostly on prior knowledge integration, we have the omic data, but now we have not only the prior knowledge integration, we also have different, let's say, modules that we introduce to this particular framework. We can integrate observation covariates onto the latent space. For example, let's say you have patients with different age groups, and you know that patients that, let's say, are older have a higher risk of not surviving the disease. So you'd like to kind of account for this particular source of variation such that it doesn't confound your survival information in the end. So you want to actually know the reasons why patients exhibit high or low survival rate based on biological pathways and not based on, let's say, age, which you have measured already. Another thing that we've introduced is integration of spatial and or temporal information based on a GP prior, which makes the assumption that samples that are close to each other, that, for example, cells that are close to each other, they are expected to also be similar to each other in the latent space that is inferred by the model. Then we also add other modules like non-negativity constraint, because as you know, single cell data, they consist of usually count data and then undergo some sort of normalization or log transformation process, and you end up with non-negative data with lots of sparsity. So in order to account for this excessive zero inflated sparsity, we introduced a non-negativity constraint. So basically the results are even more interpretable because essentially they are bounded by the zero. And also for the different omic data based on their statistical properties, we allow the user to use different, let's say, likelihoods. So you can use Bernoulli likelihood, binomial distribution, zero inflated binomial distribution, Poisson distribution, and so on. So that is left up to the user based on the data that they provide. So this is not, let's say, restricted to only single cell data or patient data. So now I'd like to focus on two main studies that we applied MOFA-FLEX on to kind of showcase some of the features and functionalities of this particular method. We applied MOFA-FLEX on a large-scale multi-omic site-seq dataset, which profiles immune cells from spleen and lymph nodes in mice, so in murine. So we try to then integrate the transcriptome view with a second view quantifying the expression of surface proteins. And the data has been essentially sampled in two different time points. So there's a bit of a batch effect included. So what we do is we introduce uninformed latent factors in order to sort of capture technical variation. You can think of it as let's say, principal component 1 and 2 in the sense of PCA. And then we inform the model with typical general gene set annotations from the hallmark, as well as cell type specific signatures in order to capture different cell types present in the data. After removing the first two principal components, because they capture, let's say, the different in the batches. We perform a UMAP computation of the data onto the two-dimensional latent space and see that the batch effect is pretty much non-existent, while at the same time, based on the information coming from the latent factors informed by the different cell types, we preserve the information coming from, let's say, the cell types. And this is also visible when we isolate different pathways coming from the specific cell type annotations. For example, we have here the marginal zone B cell pathway or the gene set, and this particular factor assigns a very high, significantly higher score to the B cells compared to the other cells. The same thing goes for the T cell pathway that assigns quite high factor scores, let's say, to the CD4 cell, T cell. We have the same for dendritic cells, natural killer cells, monocytes, and so on. And again, after kind of seeing this pathway level decomposition of the data, we can then focus one layer deeper and have a look at the actual factor loadings that describe each of these latent pathways. Now we see here for the RNA, for the T cell, that there are particular markers like TRBC2, BCL11B, and LEF1, which are quite well known and correlating to T cell structure. And we see in the protein, which is the modality that had not been informed a priori by the T cell signature, that the main marker is the CD4 marker, which is also correlating to this particular CD4 cell type. The second use case is when we apply MOFA-FLEX on spatial transcriptomics to kind of tease out the pathway activity in breast cancer patients. And we applied the method on two groups. So we have the xenium group with around 300 genes, but providing spatial information from adjacent FFPE slides. And we also have a high coverage chromium group, which has more than 3,000 genes. Essentially, this serves to inform the model with particular biological pathways. Again, we inform the model with gene set annotations coming from the hallmark, so that's why the H here, to try to figure out, okay, what are the main drivers of variation in this particular data set? And we see here that estrogen response is highly regarded as the most important in this particular data set. Then we have androgen response, apoptosis, then again estrogen response late, apical junction, and so on. When we have a look at this particular pathway modeling the estrogen response, we see that it significantly enriches across non-invasive ductal carcinoma samples and invasive tumors. So basically all these cancerous cells, they are highly enriched, basically having a much higher estrogen response compared to the other non-cancerous cells, let's say. Next, we can inspect the composition of the, so basically have the canvas of the data and try to paint the picture where this pathway activity is mostly focused on. And we see here that for the invasive tumor, it is mostly focused on the, so it has a much higher activity of the, let's say, estrogen response. And then again, we inspect the composition of the estrogen response, early pathway, in order to see markers that are potentially relevant in the diagnosis and the prognosis of breast cancer. And among the, let's say, the annotated markers, we also see different markers like FOXA1, GATA3, TFF1, and TFF3 that are estrogen-regulated markers associated with luminal breast cancer subtypes. In addition, we also have CCND1, ERBB2, which are associated with cell proliferation, metabolic rewiring, and generally poor prognosis in breast cancer patients. And in particular, the ERBB2 receptor, also referred to as the HER2 receptor, correlates strongly with, let's say, the aggressiveness of the cancer and is strongly associated with poor prognosis in patients with breast cancer. So in summary, MOFA-FLEX is a factor model framework for integrating omic data with prior knowledge. So I think this is the main takeaway from this presentation. If there are two aspects that you should remember, is that we are able to integrate prior knowledge from gene set annotations in order to give the model a sort of a biological context coming from biological pathways. And the second is that MOFA-FLEX is, let's say, the current state-of-the-art framework to integrate all these omic layers, including, as I said, domain knowledge. We can provide different, let's say, modules to accommodate and tailor the analysis to the data set and to the requirements of the analysis of the user. We can integrate spatial temporal information, which leads to smooth pathway scores across tissue and time. As I said, samples that are close to each other, we expect them to have a similar kind of pathway decomposition. And again, MOFA-FLEX is now an open source package, which is compatible with the current software ecosystem or the universe. So finally, I'd like to thank the European Research Council for providing resources to fund this project. And I'd like to thank my colleagues, especially our team lead, Florian Büttner, which has been very helpful in this project. Also my collaborative partners from the Stegle Lab, Oliver Stegle, Martin, Florian, and Ilya. And the software, as I said, you can take a picture or scan this QR code or send me an email if you have any more questions about the software. But yeah, this is it more or less. I'd like to thank you as well for your attention. And yeah.
Okay, thank you, Arber, for the fascinating talk. And I'd like to invite any member of the audience to ask questions to Arber about the presentation.
Maybe I can start by asking a question. So I have used MOFA, like the traditional MOFA before, and I noticed that the required data size is quite big. So in with this MOFA-FLEX, what is the minimum size of data that you would need to have? I noticed that you had quite big data sets in your examples.
So yeah, that's a good point. So there is a certain minimal amount of samples required for sure, because these models, they usually span across hundreds, if not thousands of features. So you usually end up in a sort of a P larger than N issue. We have applied this method on data sets as low as 50 samples, and lower than that, I think you might end up in some really not meaningful decomposition, let's say. These are methods that are data hungry, not as data hungry as deep learning methods, but let's say a few hundred of samples would be optimal. But as I said, I think it can probably go as low as 50 samples. It then depends on the kind of the properties of your features. If you have too many features, then or too many latent factors, then you might end up in a really sparse latent space. But I think it can be tweaked. So maybe to answer your question, probably 50 is a good number.
So, I mean, I cannot see myself, but I guess you see me, right? So
yes, yeah.
So my name is Xing. Nice for have you here. So, Arber, right? So actually, I was in Germany for a very long time, and I just come to Finland to start my lab 2 years ago. So I also know MOFA. Actually, for your talk, I have some, I can imagine due to the limited time, you didn't go details. I have some specific questions about the technology. So one question is like, you have so many different data sets, right? But from biology
Hmm.
perspective, for example, you have transcript data, you have protein data, you can even have mutation data. So
Yes,
biologically, these features, they are linked. So probably a gene can have
you
mutation
are right.
and have isophones. Is
Yeah.
this considered in the method, or how was this considered in the method?
So one thing that I can say that seems to work fairly well is that the decomposition allows for different sets of latent factors. So each factor can be either, and you probably know this from MOFA, it can be private to a particular view or modality. So it can be only focused on the transcriptome and explain variants only in the transcriptome, or it can be shared across different modalities. So that can be one particular factor, like this one, androgen response. It's kind of shared across the RNA and the proteome.
Mm-hmm.
And based on correlation structures, then you can see that particular markers, like the ones that are highlighted here,
Mm-hmm.
they show a strong correlation. And you can somehow, in your analysis, kind of tease out these correlations if you don't have this information beforehand. But usually what we assume, essentially we assume that your different modalities have different sets of features. So you have one
Mm-hmm.
transcriptome with genes, and you have one proteome with proteins, and you have one mutation with mutations. And okay, so let's say our main assumption at the beginning is that they are independent or different
Yeah.
features, although we
Mm-hmm.
already know that there's quite probably a big overlap there as well.
Okay, thanks for
But
clarifying this.
based on the shared factors, then I think you can
Mm-hmm.
somehow come up with
I
different
see.
correlation structures.
Yeah,
Yeah,
of course, you can probably, for example, to calculate the scores for individual features, and then later on
yeah,
you can, find
exactly. That as
approach
well. Yeah,
to
yeah,
aggregate their contribution, right?
yeah.
Thanks. So another question I have is about this training. You mentioned that when you are training the model. So
Hmm.
what is your objective function here? What are you training your model for, and and how did you train it? What's the aim of this training? I don't get
Yeah.
it. Yeah.
So let's have a look at maybe this slide. So let's say we have your data, and then
Mm-hmm.
we'd like to sort of perform this decomposition in order to explain the data. But the main kind of loss objective, like statistically and in machine learning, what's being optimized is the root mean squared error between this particular modality and the
Mm-hmm.
product of these two factors. So you take
Okay.
the joint latent space, you multiply it with the corresponding factor loading of the transcriptome, then you
Okay.
try to come as closely as possible to your observed transcriptome. And then you do the same thing for, let's say, the chromatin
Okay.
mutations and so on. So you
Okay.
kind of try to optimize this, but at the same time, being or adhering to, let's say, your prior annotations or your context, such that the factors are no longer called factor 1, 2, and 3, or principal component 1, 2, and 3, but rather here's hypoxia,
Mm-hmm.
here's androgen response, here's, I don't know, calcium signaling pathway, and each of these, let's say, latent factors corresponds to one particular pathway.
Okay.
Yeah,
I see. Thanks. So somehow you try to reconstruct the raw data, right? Through
yeah,
your
exactly. Yeah, yeah,
yeah. So
yeah, yeah, yeah.
like a autoencoder does, right?
Exactly,
Yeah.
exactly. Yes,
Yeah.
exactly.
So here I noticed that when you are integrate your prior knowledge from the database or gene sets, you have
Hmm.
these categories R, H, K. And how was this done?
Oh, yeah. So these are
Hmm?
different, let's say, sources of gene set annotations. So from
Okay,
different
so this
studies,
represents
they have,
reactome, hallmark, or
yeah,
cake.
yes,
Okay,
yes, yes,
that's
yes, yes.
that's the meaning of
It's
it.
only it's only
Okay.
the abbreviation. Yeah, exactly.
Okay, so I have some more questions. Maybe
Oh, yeah, go
just
ahead.
to finish it before others starts. Another thing I realized that in one example you said that you have those factor. So these are factor 4, factor 12, but they don't have a biological meaning is because they are like something. Why they don't have like a biological entity or meaning behind these
Yeah.
factors? Hmm?
So there's that's actually a very good question. So there are a few differences. So the model in itself can also be trained in an uninformed fashion. Like
Hmm?
because you have experience with MOFA, then you know MOFA doesn't really take these prior information. MOFA, as you said,
Hmm?
learns factor 1, 2, 3, and 4, and so on.
Okay.
What we try to do is we develop this method that takes the prior information, but then we try to integrate both. So we can have the best of both worlds, more or less. We can have a subset of factors that have been
Hmm?
informed a priori by particular gene set annotations. So we have, let's say, 10 factors from Hallmark, 10 factors from Reactome, and
Okay.
5 factors without any sort of information. And these factors that have no prior information, we can call them uninformed or unconstrained.
Okay.
They usually kind of function as the principal components or factor 1 and 2 in the MOFA, which usually explains the most variance in the data. Now, there's also another thing that may lead to having a factor 1, 2, or like
Hmm?
annotated factor, and that is after training the model, if you provide biological information, then we perform statistical tests to test whether the factor is still significant to the prior annotation. So let's say we informed one factor with hypoxia, and we know the gene set that
Hmm?
kind of tells you what hypoxia is active, but then we see whether the main factor loadings, like here,
Hmm?
whether the majority is still part of the prior gene set. And if it's not, then it's deemed insignificant. There's a called PCGSE. It's a method, a statistical method to test whether your inferred factor still resembles the prior annotations. And if it's not significant, then we rename the factor to
Hmm?
like an factor 5 or 6 and so on.
Okay.
Like you can see here, factor 4,
Hmm?
it was it had a different prior annotation, but just did not match. It was not significant. So we renamed it basically to factor 4, and it's uninformed. Because most of the factor loadings in this factor score, they are usually red. So basically, they're mostly inferred and very few annotated. But they are still useful for the method and the analysis.
Okay. So one more probably question is like, when you are dealing with these features, some are
Mm-hmm.
inferred, some are annotated. But of course, in biology, the most annoying things like you find those contradictory evidence. So I mean, have you? I don't think you probably have a perfect solution, but you can explain us how do you deal with this kind of thing.
Mm-hmm. So one thing is the method can really accommodate noisy annotations. So we do not expect, let's say, all the genes in the gene set to be active, and we also expect some of the genes outside of the gene set to be introduced to the gene set. So, for example, you see here some of the inferred genes have been added. Another issue can be there can be gene set annotations or different gene sets that have a big overlap. Like, let's say we have 2 gene sets, and out of 100 genes in each gene set, 80 are shared. We do a pre-processing step where we try to find gene sets that are similar to each other and kind of merge them together
Mm,
so that we avoid latent factors that are highly correlating, but at the same time redundant in what they explain. So because the method is a Bayesian method, all this serves as a kind of a prior distribution um, of your expectation, of your belief. And then based on the data, so the likelihood of the data, it will shape your prior information, your prior context into your posterior context, let's say. And this is what you see here. And if it's justified from the data enough, so if there's enough evidence from the data that the prior annotations are not correct, then you will see it from the model. So the model will communicate to you if your prior context is suitable or not for your data.
mm. Okay, cool. Yeah, so for the last question, I will finish. So of course, there are many methods for this kind of analysis to identify the um, important features or genes, or even prioritize them. So when you publish your paper, I guess it's published, yeah, for a while.
soon.
Did you do any benchmarking, or or how does this perform in comparing to other similar methods? And
Yes,
what data
that's
set you use
actually
for
a
this
good point.
benchmarking?
So I skipped one slide because it was basically exactly what you meant. It's benchmarking on different methods. So we have XBMAP, Spectra, and
Mm-hmm.
MOFA-FLEX is our method. And so basically, briefly, we can summarize the benchmark in this particular plot here, where we show that both the reconstruction error of the data and the reconstruction error of the prior annotations is highest for our method compared to the other methods. But this will be is part of the paper and will be part of the published paper. Yeah.
Okay. Okay. Thank you. Thank you for clarifying these
Yeah.
details. I think it's
Thank you for
very
the
interesting.
questions. Really interesting.
Yeah. Thank you.
Rishi, you can.
Yeah. Thank you. Like great talk and yeah, really good questions from Julia and Zin. Almost like Zin has taken my many of questions you have already asked. So I have one question. Like you asked, like that the batch correction you have done by removing
Yeah.
the principal components.
Yeah.
So yeah, that is quite interesting. So you found like principal component one and two, and they are affected by the batches, and you use them. You just remove them to rectify it. Have you tried in different data set, or it only worked here?
So we have tried this on a different data set of patient information from DLBCL patients, so large lymphoma B cell. There we even went one step further and tried to kind of correct for different sources of variation that we were not interested in, like stromal infiltration, proliferation, because we knew that we were interested in finding patients or subpopulations of patients that exhibited bad survival. And we knew that particular biological pathways or biological processes, like, as I said, in proliferation and stromal infiltration, they would lead automatically to, let's say, bad survival. We removed those as well, and we were able to focus on other, let's say, sources of biological processes that may lead to, let's say, bad survival. I have to say there are lots of methods that do batch correction, especially methods that they accept a sort of a indicator. This is from batch 1, from batch 2, from batch 3, and they perform quite well batch correction. I have to highlight that for this method, we did not provide that information to the model. So the model had no idea which batch the sample belonged to. And that is a side effect of having two subsets of factors, one subset of factor focusing on the biology, and one subset of factors focusing on, let's say, technical variation or other variation that is not accounted for by the biological context. And in this case, the uninformed factors, they focus mostly on, let's say, the main sources of variation, which came from the two batches. And after removing those, then performing the, as I said, the UMAP computation on the latent space only from the biological pathways, then you no longer see the batches, but you preserve, let's say, the biological information coming from the cell types.
Okay, yeah, great. And yeah, one I probably haven't understood well that in the third slide, the second plot, I think when
This
yeah, the
one?
second yeah, what are the x-axis and
Yeah.
y-axis, and it is not UMAP plot or
No, it's a simple scatter plot. So
okay,
if you go
yeah,
back to this page, then we have a different patients here. You can think of different
yeah,
patients and the, let's say, the RNA of the patient, proteome, RPPA, and so on. Then
yeah,
we focus on this particular component where we have,
Okay.
again, the patient, but the patient is now described as a vector of different pathway scores. So let's say here we have melanogenesis, here we have androgen response, and so on. So what we do is take all the patients from the different cancer subtypes and see where they end up in this
Okay.
particular scatter plot. So androgen response is the pathway, and these are the factor scores for androgen response. You see that the androgen response pathway assigns a significantly higher score to patients with prostate cancer compared to the patients without prostate cancer.
Okay.
And you can read this as androgen response being elevated significantly in patients with prostate cancer. And that's more or less the idea. And the same goes for melanogenesis for skin cancer.
So this kind of plot like easily can be plotted with MOFA-FLEX.
Yeah, yeah.
Okay.
So you can
Good.
take exactly two different pathways and then plot the scores and color them by different metadata in your data set.
Okay, great. Yeah. Yeah, I don't have any other questions. They has been asked by Zina or
No,
Julia.
thanks for the questions.
Yeah.
Okay, I might ask a very general question. So, maybe to conclude the session of questions. So I understand that so your package is intended to be used by biologists and medical researchers who don't necessarily have an extended statistical background. So how do you advertise MOFA-FLEX to these people, and how do you explain to them so you get them to use it shortly?
That's actually a good point. So we have, after we developed this software, we then obviously also took some time and prepared some really nice documentation of the software package and made sure that we have really step by step different analysis of different use cases, especially for MOFA users, like some of you here. We have taken those, let's say, notebooks or analysis that are online and tried to emulate everything, kind of mirror the results again, but using MOFA-FLEX at this time. And another project that will be coming maybe soon, in the near future, is we will be writing kind of an agent or a large language model based method in order to basically serve as an interface between biologists and medical professionals and our code, so that you basically get a questionnaire about your data sets. You get asked, okay, what's the data set about? Can you explain more about the data set? And in the end of this questionnaire and kind of interacting with the agent will generate a very well suitable and maybe a tailored configuration of the method for you, because it can be a bit intimidating having all these different modular pieces and having to kind of combine them together. So that might be something in the future. Yeah.
Cool. Okay. Well, if there are not more questions, thanks a lot, Arber, for the presentation
Thank you for your
again.
time.